Today we're going to start chapter 20. Hopefully we're going to give ourselves about 2 weeks for this chapter. We're going to take a test on this before spring break and start fresh when we get back in April. So we skipped a few chapters about evolution and taxonomy, and now we're going to go over viruses, bacteria, and archaea. So 20.1 is about viruses, viroids, and prions, okay? All kind of stuff about, we're going down the virus road, per se. All right, so what is a virus? A virus is a non-cellular, so that means it's not a cell, parasitic agent. That means it's a parasite, right? It's going to take something from the host. And it consists of an outer capsid and an inner core of nucleic acid. So it's non-cellular, it's parasitic, it has a capsid, and it has nucleic acid. Okay, those are the things that I took from that sentence. So far, there's 2,500 species identified, and this book is a few years old, or actually it's probably about 8 years old, so there's probably way more than that now. They have some characteristics of living organisms, but they're non-living, okay? These guys do not live on their own. That's why they invade your body to use your machines, pretty much. Okay. They have DNA or RNA, and they evolve and replicate like something that's living, okay? But that's it. They just have some characteristics. They're not living at all. But, it's a big but right there. They can only replicate using the host cell's machinery, okay? Why? Because they have no metabolism, they do not respond to stimuli, and they do not fossilize. Again, they're non-living. They have to use your cells or a host cell's machinery. They have to use your ribosomes. They have to use your rough ER, your smooth ER, all of your organelles, your blood, your travel system, transport system throughout the organism. They have to use all of that stuff. They don't have any of that. So that's essentially what a virus is, and we're going to show you some pictures. I know that was a lot of writing. I'll give you just a second. All right, so here's some pictures of some different viruses. You can see the orange outer capsid or capsule, and then it's showing you the DNA, the nucleic acid that's inside. And the reason your book says nucleic acid instead of just DNA is because some viruses have RNA. So they didn't want to say either or. So they are both a nucleic acid. So that's why they use that terminology. Here's another virus. It almost like sits on the cell, sits down on it almost, and injects its nucleic acid or its DNA into the cell to start to be replicated. A long time ago, somebody said this looked like something from Chicken Little. I don't know if you guys are too young to know what Chicken Little is. I never watched it because I was a little too old, but if you know what they're talking about, then maybe that'll help you out. There's another virus showing you the protein molecule and the RNA core. There's a little bit more of a complex virus. This looks like a toy to me. You can see the nucleic acid inside, and you can see the purple capsid or the blue capsid, and then it has even another layer that's like a lipid layer, which means it's a fatty layer on the outside. And you have all of these little receptors. And so when it lands on the cell, those receptors match up with the cell's receptors, and it almost tricks the cell to think it's a good guy. So let's look at this structure. of a virus. Viruses are categorized by three things. I'm not loving this orange here. Size and shape, the type of nucleic acid, because the type of nucleic acid tells us if it's double-stranded or single-stranded, right? Because which one is which? Single-stranded is RNA, and double-stranded is DNA. And then finally, the third thing is the presence or absence of an outer envelope. Those three things determine how the virus is categorized. They all have at least like an interior capsid and something inside their core, like RNA or DNA. We've already talked about that. So here's some stuff that you need to know. If a capsid is the outermost structure, that means there's nothing else on top of it, okay? Like this one has the capsid that's in blue, and then it has another layer on the outside. So this would be categorized differently than this guy, okay? So if the capsid is the last thing on the outside, it's called a naked virus. It means it's almost like it doesn't have any clothes on. And then if it's surrounded by an outer membrane or an envelope on the outside, it's called an enveloped virus. It has clothes on. So let's write that. No clothes. And then clothes on. That's what it's like, okay? You have naked viruses and you have enveloped viruses. So these guys are parasitic in nature, okay? They cannot replicate outside of a living cell, but they have a genetic material that needs to be replicated. So therefore, they must infect a living cell. How do they do that? The virus hijacks the cell's protein synthesis machinery, like I was telling you about. You know all that stuff. You know that the RNA, if it's DNA, it's going to be read. It's going to be transcribed and, translated using your ribosomes, your Golgi, your rough and smooth ER, your vesicles, everything like that. That's what it's hijacking, okay? So that protein synthesis machinery is what it needs to replicate whatever nucleic acid it has. It can be RNA or DNA and any other parts of the virus that, needs to be made. So because it hijacks these things and it literally goes into overdrive and makes 1 million of these little guys, some infected cells are killed or damaged by the virus. Just because they're so vicious. So an example of those would be pneumonia or bronchitis. Those are some viruses that cause those things. Some bacteria can cause that too, though. So host specific. I'm sorry, I thought my door was open. So what kind of host does it like? Okay, specificity. So particular viruses, or a particular virus is only capable of reproducing in a specific organism. For instance, they are host specific. For example, like rabies can only be infected or only use animal cells. They can even be tissue specific. It can be organism or it can be tissue specific. Those examples are hepatitis in your liver and HIV in blood cells. You don't hear, oh, I got HIV in my toenail, or oh, I've got HIV in my of my skin. It's in your blood. So essentially, it's everywhere. So remember, it's determined by the structure of what it likes, like if it's, or if what it's specific to. It uses a lock and key manner. with the host surface. So I was telling you up there about the receptors. It's the same thing. Some medicines mess with this to prevent further infection. So these viruses land on these cells, and the receptors communicate like a lock and key. It almost allow it inside the cell, so it tricks it. So some medicines will alter the receptors on a cell for a period of time while you take that medicine, so that when that virus lands on it, it won't lock and it won't connect. That lock and key won't work. Therefore, that that virus can infect that host, and you get better over time. So the reproduction of viruses, this is a big thing for this chapter. You have 5 steps. Number 1 is attachment. The virus has to attach. It attaches to the surface molecules and the host cell receptors. We've talked about that. This is the 3rd time we have said, or I have said, cell receptors and the surface. Okay? And then after it's attached to the host cell, it's going to penetrate that outer layer of the cell. So there's 2 options, okay? The host cell can eat or engulf the virus, or the virus can inject its genes or genome, or if you want to say their nucleic acid, you can. So it's either going to swallow the whole virus, or the virus is going to just almost like a needle, it injects its DNA or RNA into the cell. There's 2 options there. The 3rd step is biosynthesis, and this is where you're going, it's going to use all the cell's parts. It's going to use all those protein synthesis machinery parts. So it says viral components are synthesized using the host stuff. This is the 3rd time we've said that too. It's using all of those parts that we've already learned about. How your body or how a living organism makes proteins. It's going to use all of those parts. So after the parts are made, then you have to put it together. So maturation is the assembly of the new virus. So this is going to where it's assembled. Okay, put it together. And then, of course, the last part is the release of the viruses that your body or the living organism has made. So it says new virus exits through lysis or budding in order to infect new host cells. So it's almost like, have y'all ever seen a pregnant spider? Have you ever stepped on a pregnant spider? That's kind of what it's like sometimes. It's like a burst of, the cell ruptures. It explodes almost. Think about if you had one of those little slime balls and you squeezed it so hard that it busted out the top. That's what it's like. So it kills the cell. So those are really important. Big deal, big deal, big deal. So I think I want to stop there. I know we're not finished with 20.1, but I don't want to give you so much information to where you're not going to remember what we talked about. Okay, I only want you to know the stuff about this type of virus right now. I want you to know what a virus is, how it's categorized, how it can be specific to certain things, and how it reproduces, how it attacks the host cell or uses the host cell. Okay? Because its goal is not to kill the host, because it needs the host. Sometimes we think of viruses as being horrible because they affect us in a negative way, because they are parasitic. But those little guys, they're just trying to survive too. they don't mean to kill the host, because then that means they have to go out and try to attempt to find another one. But they're just trying to live like we are. All right, so we're going to stop right there for today.